thank you very much for being here. Thanks, Muriel, for inviting me to this nice School of Astrobiology. It's a very good opportunity to meet again some of the friends I have in this community. And okay, my background is biochemistry and molecular biology. I'm a member of Center for Astrobiology since year 2000, and I lead there the group of molecular evolution. Okay? So, we have been talking about all this stuff, okay, from the evolution of the universe to the formation of a rocky planet with atmosphere and water. So, thanks very much, Daniele, for bringing the water, because now life can emerge. Until your talk, it was impossible. Now, life can emerge. My presentation will deal with this phase of the evolution, so the origin of life, an issue that is, as you know, plenty of very relevant questions. When did life evolve? How? Was a product of chance and or necessity? Where, in particular, on Earth or in extraterrestrial bodies? And if it appeared once or several times? Okay? So the last two very nice presentations have taken us from this picture to this one, which is not so bad, okay? But we have to move to this one, to the present. Okay? This is a picture I took yesterday. Fortunately, because now it's raining, so this is good, because you bring the water. And, okay, so now we are surrounded by life. We are members of an amazing biodiversity. So the question is how life originated, how life evolved until the present situation. If we summarize some of the results my colleagues have been showing you in the first slides, we can say that the origin of the Earth and Moon dates back 4.57 gigayears. Crust and oceans represent at 4.4. The oldest sedimentary rocks could have this antiquity, 3.9. The isotopic fingerprints of early life, 3.85 and perhaps 4.1, it's not clear as far as I know. And the oldest fossils are here. For sure, 3.5 and perhaps 3.7, okay? Okay. So, life could have started to try to emerge here. Some chemical processes leading to life, probably. As we heard yesterday, this late heavy bombardment probably sterilized the whole planet, but chemical species produced here in that era could have survived the sterilization process. So it could have been very nice starting products for new experiments. Okay, and life definitely has had this 350 million years to originate, because here we have life. Okay, so how we study this this part of the history of our planet? We use two approaches. The first is called the bottom-up approach, or from the past to the present, on from the non-living. This is the field for astrochemistry, geochemistry, prebiotic chemistry, and also synthetic chemistry. Okay, and we have the opposite approach, the top-down approach, or from present to the past, or from the living. Okay, this is the typical way paleontology works, also microbiology, phylogenetics, and the models for minimal life. Okay, and they have to reach the same time, more or less, the time corresponding to the origin of life. Okay. In fact, the Earth was a global laboratory once water was present on our planet. We had sources for atmospheric synthesis of several gases, also extraterrestrial delivery of organics. We have been listening some data about that. It could have brought some organics here necessary for making this special kind of chemistry called life. Also aqueous phase chemistry, okay, hydrothermal and geothermal synthesis in and hydrothermal vent scenarios are perhaps interfacial chemistry. All this could have been operating together, interchanging part of the solution and making this global experiment that led to life. Okay? The first known fossils are fossilized forms of what we call at present microbial mat, layered disposition of microbes, and this is the oldest stromatolite recorded to date. Is sorry, it's quite different from mine, so it's a bit difficult for me to. Okay, these stromatolites were found in Australia, Pilbara Formation, 3.5 in rocks of 3.5 gigayears ago, and okay. A couple of months ago, this paper was published that claims that these formations here are also stromatolites in rocks of 3.7 gigayears old. It's not clear. Much has to be done on these samples, but for sure, or most of the community agrees that these are the remnants of the first life on Earth. We have also microfossils. Individual cells could be distinguished here. This is the famous Shop's paper in Science. They claim that this morphology could correspond to cyanobacteria. It's not clear at all because it's only a morphological evidence. And we have also these very nice fossils from Wasai et al. That are a bit younger, but also very interesting proofs that life has originated at that era. But we have to be very careful, because if we say that life was present here because of the morphology, we have to have in mind that chemists are able to make structures that resemble very much these microfossils, and they are formed only by silicates and carbonates, so no life there. So they are called abiotic biomorphs, and one of the main scientists in this field is my colleague Juanma García Ruiz in Spain, in Granada. So yeah, danger, be careful. Morphology is not a definitory sign for life, okay? The composition of life. Again, the periodic table of elements, we are scientists, we have to show this from time to time. And what I would like to remind you is that these 6 elements, as all of you know, account for the 99% of the mass of all living beings. Okay? We have also other compounds, trace elements that are necessary for life, but only represent the 1% of the mass of living beings. Okay? And here's an example. You can see the composition of our body. Okay? You are mainly oxygen, carbon, hydrogen, and nitrogen. And that's all. If we compare our composition with this similar picture was presented the first day of this school. The stars are mainly formed by hydrogen and helium, and this is what you astrophysicists call metals. It's quite funny because carbon is a metal for you. Okay. It's okay. It's okay. You have invited me. You have invited me, so I would say that carbon is a metal. Okay, but only in this slide. Huh? The crust of our planet is mainly made of silicates, so oxygen, silicon, aluminum, iron, etc. But organisms have a quite different composition. Okay, I keep the code of the colors, and so there's oxygen, carbon, hydrogen, the other 3 main elements, and the other from 1 to 2%. Okay. So we are these elements, because we are mainly water, because water is, as we were discussing before, essential for life. Is the best solvent. Sorry, it's crazy, it's not so well designed. Okay, it's a very good solvent. It allows chemical reactions, a lot of them, and it gives thermal stability to the environment. Okay. And also we are carbon. Carbon is a very peculiar element because it allows a complex chemistry based on covalent bonds, so sharing electrons, and it makes different aliphatic and aromatic structures. So, again, forming functional groups with the other 5 main elements of life, it is the basis of very complex chemistry, the complex chemistry that is required for life. Okay? Then, okay, so we have elements, water. What else? Okay, these precursor molecules, with from 2 to 5 number of atoms, it's okay, quite small. Then we have low molecular weight organic compounds, those that appear in your analysis when you go to the doctor because you have eaten a lot and so on. Cofactors, antibiotic, hormones, etc., etc., etc. Then monomers, this is definitory for life, such as nucleotides, amino acids, sugars, and fatty acids. And then the main molecules of life that are biopolymers, DNA, RNA, and proteins. And you know that the flow of genetic information in nature goes from DNA to proteins, DNA replicates, DNA transcribes to RNA, and RNA is translated to proteins. Okay? This is working in all cells, and also in some viruses, RNA replicates, or some viruses have proteins that allow RNA to replicate, and RNA can be retrotranscribed to DNA. So this would be the overall picture of the flow of genetic information in our biosphere. Okay? Then we will talk a little bit about the point that viruses can be considered or not as living beings. This is another issue. Okay? But this is the overall picture. Okay?
Do those colors mean anything?
Those colors mean nothing, basically. Yeah, but they are nice, okay? These colors are the typical toy models for chemistry. So each element has a definitory color. For instance, nitrogen is blue and hydrogen is white.
But not for ours.
Not for others. This allows you to trace how the helix of RNA, okay, goes through through this complex molecule, and here the colors have been chosen to define different domains of the protein. By the way, this protein is hemoglobin. Okay. So these are the main molecules of life. If we continue increasing the complexity, we go to the cells. Okay. In nature, we have 2 kinds of cells: the prokaryotic cells without any internal organization, let's say, and eukaryotic cells with a defined nuclei and lots of organelles and higher morphological complexity. Okay? Prokaryotic cells account for bacteria and archaea, we will see them later, and eukaryotic cells for eukaryotics. But what is life? Life is the composition of life, or life is something else? This is a quite complex question. We could spend the whole day or a couple of days talking about, but we are not allowed, okay, about what is life. Dozens of scientists and also philosophers, of course, have been thinking about the definition, the possible definitions of life since Aristotle. Okay, this is a nice timeline made by my friends David Moreira and Puri Lopez Garcia, and you have some of the definitions of life from Aristotle to now. It's quite interesting. The definition that Gerald Joyce coined is a definition that says that life is a self-sustained chemical system capable of undergoing Darwinian evolution. It's not too bad. It takes the main components of life in almost one line. Okay, so chemical system, you need chemistry, self-sustained, without self-sustaining there is no life, so metabolism should be there, and capable of undergoing Darwinian evolution, because everything in nature evolves, as we will see. It's also interesting the definition of my friend Kepa Ruiz-Mirazo, and also co-worker, that they say that life is an autonomous system with open-ended evolution capacity. Okay? So, autonomy and evolution. This is what defines life. At the end, life is defined by the combination of a replication machinery, of a metabolism that allows living beings to interchange matter energy with environment, and a compartment, something that defines the living being. on the outside. Okay? The combination of these three features allows life. Okay? So life is a combination of these. How we classify life? Okay, it's an interesting issue because we are surrounded of different kinds of living beings. Some are microscopic, so we have to use the microscope to see them. Others are macroscopic, and we are part of the biodiversity. We are not more evolved that part of the biodiversity that surrounds us. And we have found life also in extreme environments all over the planet. Okay, so if we pick samples of all these environments and we try to classify life, we find, okay, microorganisms. This will be bacteria and archaea, we'll see. And I have chosen these 4 examples of eukaryotes. Okay, so we have a plant, a fungi, and 2 animals. Okay, by the way, this is the best representative of our species. Okay.
Which are the Darwinian?
Yeah, that's right. That's why he's here. And it's a biologist, not an... Well, it was a geologist, as you know. It was formerly a geologist, but okay, he discovered biology, and okay, he couldn't he couldn't go back. Okay, so Mr. Charles Darwin. If we compare the organization of these systems, bacteria and archaea have a prokaryotic nature, and all the eukaryotes are formed by eukaryotic cells. Good. If we compare genetics, all of them are the same. Are DNA, RNA, proteins. Okay? If we compare metabolism, the core of the metabolism is common to all living beings. So, the conclusion is, could all of us have evolved from a common ancestor? Okay, this is the most parsimonious answer for the fact that we share a lot of features. The main features of life are shared by all representatives of life. Okay, the answer is yes, we share a common ancestor, because when we were able to compare the genes and then the full genomes of all the species around us, we were able, Carl Busch and Norman Pace were able to construct these wonderful phylogenetic trees with 3 domains, archaea, bacteria, and eukarya. So the 3 main domains of life are there, and a point, okay? That point would be the common ancestor, okay? And they have emerged from that point in some way. We can think about that. But what is interesting is to recall that the last paragraph of On the Origin of a Species by Charles Darwin, the best book that was written by human in the history of humanity, perhaps. I'm a bit fan of Charles Darwin, as you can see. He says, there is grandeur in this view of life, with its several powers, having been originally breathed into a few forms or into one, and then evolve. Okay? So, in 1859, he was able to imagine that all life has evolved from a common ancestor. 130 years later, it was proven that was the case. So, chapeau, Mr. Darwin.
Wait, he said a few forms or one. He didn't say just one.
Yes, and then, okay, of course, he was cautious. Say, okay, just in case, just in case. And also, you know something that is quite interesting in the history of science. This is the last part of the first edition of On the Origin of the Species. Then he was forced to introduce by the creator here for the second edition.
By his wife?
Yeah, among others. Yeah, Emma Darwin was a very active person. So from the second edition to the end, originally breathed by the creator. Okay, originally breathed. I prefer the first edition. If we change a little bit the topology of the tree, and this can be done using phylogenetics, we get this shape with the 3 domains, bacteria, and archaea and eukarya being sister groups, okay? That would be the last universal common ancestor, okay? Our former ancestor, and that would be the origin of life, in fact. So the origin of life has nothing to do with Luca, which is at the top of the common trunk of the tree of life. The origin of life is at the roots of the tree. Okay? So this is quite interesting. By the way, this topology of the three main domains of life can be changed if you change the gene that you are using to make the phylogeny. Okay? So you can have Archaea and Eukarya as sister group, or Bacteria and Archaea, or Bacteria and Eukarya, or the three groups evolving from a common point. Okay? Most of the genes give this topology, that's why we use mainly this one. Okay? In any case, the take-home message is that tree is not something like this, two branches, not eukaryotes and prokaryotes, but something like this, three branches. It took a while to find these pines in Spain, okay? But I think they are, in particular this one is, for me is wonderful. It should be preserved for eternity. So, bacteria, archaea, and eukarya. Indeed, this tree is quite elegant, quite clean, with very definite branches, are not a real metaphor for the tree of life. That is something more similar to a bush or to a climbing plant, okay? With lots of intermingling branches, with lots of, let's call it, horizontal gene transfer between the main branches. This is more realistic representation of the tree of life, okay? So, should be the bush of life or something like that. In fact, this is quite biological, this is quite real, okay? When you see very clean schemes, don't trust them very much, okay? This is a quite nice representation of the possible evolution of life, with a lot of horizontal gene transfer, of genes taken from an organism and inserted into other organism. By the way, this is about cells, but are viruses alive? Let's talk a little bit about viruses. Here you have a virus. This is a human immunodeficiency virus in particular. They are macromolecular assemblies that replicate inside the cells. They have a molecule with genetic information. It can be DNA or RNA. They have a protein capsid, here in blue. And some of them are enveloped because they have stolen part of the membrane of the cell they were infecting. Okay? The main features of viruses is that they replicate, they mutate, and they evolve, but they are obligate intracellular parasites. So the first set would say that they are living organisms, but the last one, they are non-living organisms. This is an open issue in biology. Are viruses alive or not? It depends on how you define life. Okay? In particular, there are a very nice controversy about the point that viruses should be or not considered relevant for the origin of cells. David Moreira and López García published 7 years ago this nice paper in Nature Reviews with 10 reasons to exclude viruses from the tree of life. Among them, that viruses do not evolve in the absence of cells because they don't have metabolism, so they need the cell, okay? In fact, all metabolic genes in cells have cellular origin, so viruses apparently don't have and haven't before cells for metabolic activities. Viruses are polyphyletic, they don't have a common ancestor as cells do. And this is also a quite philosophical question. Simplicity does not mean antiquity, and we have to bear in mind that. Okay? Other authors, yes, they say that viruses are relevant for the origin of cells, and this is, for instance, the work of Koenig and others. They think that a biosphere predated the biosphere of cells. I mean, the biosphere we can see from Luca to now. Okay, this is an open issue, but I would like to show you, because this field is plenty of interesting open questions. In my opinion, what is relevant is not what viruses are, but what viruses do. And viruses infect all organisms that we know, infect the 3 domains of life. And it's estimated that the number of viruses in our biosphere can be 10 times higher than the number of cells. So the so-called biosphere is something that surrounds the tree of life. And also, it's very important to notice that main horizontal gene transfer, an important number of them are done by viruses. So viruses are main players of horizontal gene transfer. So they are fundamental for the evolution of life. Okay? This is what viruses do. This is the important part of viruses. Okay? But let's move. Let's move towards the origin of life. A nice picture again. This was taken in Ho Chi Minh City 4 days ago. Okay, I was looking for a nice roots, and here is easier to find nice roots than in Spain, that we don't have this kind of wonderful trees you have here. So let's go to the roots of the tree of life. Okay. If we extend the common trunk of the tree of life, okay, that is the important issue for this talk. We had the precursors at the beginning, these gases we were talking about this morning, and they could be converted into the biomonomers. The first models for this, okay, the first ideas for this were also written by Darwin in his books and in his correspondence, but the first models showed up in the decade of the 20s and 30s of last century by Alexander Ivanovich Oparin in Russia, John B.S. Haldane in England. And let's focus on Oparin's stepwise stereotrophic origin of life. Okay? In his view, a reducing atmosphere, not the one we think it really was, allowed the synthesis of organic compounds, then the formation of this primitive soap. This is a very nice metaphor, the primitive soap. Then coacervates, aggregates mainly formed by proteins and with other features, and then primordial heterotrophs, so organisms. Okay, this was published originally in Russian, 1928, but it took 12 years to be translated to English and to go to the whole community of scientists. So this is the model of Oparin. But it was in the 20s. 30 years later, we moved to the experimental prebiotic chemistry, thanks to the work of these two pioneers, Stanley Miller in the States and Joan Oro, a Spanish scientist working in the States. Okay? I can imagine that in the 50s in Spain it would be impossible to make something related to the origin of life and to science in general and to culture in general. Okay, but Stanley Miller and Oro were the pioneers, okay, in this field. So they inaugurated prebiotic chemistry as an experimental science. This is the famous Miller experiment. He, because he was working alone, although he was working in Harold Urey's lab, he mixed the four gases that were considered to be present in the primitive atmosphere at that time, very reduced forms, okay, in a closed system, previously sterilized, of course, and some sparks resembled the energy that was present in the early Earth. Okay? 2 weeks later, amino acids and other biopolymers could be sampled from this primitive ocean. Okay? So it was a brilliant experiment. So the first biomonomers, or the first biological or protobiological molecules, could be formed only starting from these gases and making them react thanks to this accelerating, let's say, agent. Okay. What was interesting, because the experiment was very popular at the moment, of course, but it was very interesting that 16 years later, the Murchison meteorite, carbonaceous chondrite, fell in Murchison, that's why the name, in Australia, and when scientists analyzed the organic material in this meteorite, they found amino acids, amides, aromatic compounds, aldehydes, a plethora of different molecules that life uses. Okay? And it's interesting that if we compare the relative amount of aliphatic amino acids, in particular, in the Murchison meteorite and the Miller experiment type experiments, okay, You see here the composition in Martian meteorite in blue, and this has been synthesized in a modified version of Miller's atmosphere. Okay? So you see that perhaps Miller-type chemistry is operating in different bodies in our universe. So perhaps our universe perhaps is plenty of amino acids. Who knows? Okay? Some words about Joan Oró. He was able to synthesize adenine, so one of the nucleobases used in DNA and RNA, by combination of molecules of hydrogen cyanide. I will not go into details, but this was a very happy Joan Oró when he discovered this in 1960. So it was an alternative pathway. So Miller could explain the origin of amino acids, let's say, and Oró the origin of nucleobases. Okay? Miller was, after a couple of decades, very popular in Spain, and our former king, Juan Carlos I, named him Marquess of Oró. You have to choose a coat of arms. And he decided to include some references to his wife and their children, a mount which is very popular in Catalonia, and adenine. So it's the first molecule in the history of heraldry that appears in a coat of arms. It's interesting. Okay. It's also important, I don't have time to explain everything, fortunately, for you, but it's also interesting another model for the origin of life called the chemautotrophic theory. In this model, there are two pathways for early carbon fixation that at the end go to primitive cells. The first is the reverse citric acid cycle, so the reduction of CO2 on metal sulfides. This is the seminal work of Günther Wachter-Heuser and others like Morowitz. And there is another pathway that goes to acetyl coenzyme A net synthesis. I will not go into details. It has been promoted by Russell and Martin. These models for chemautotrophic origin of life are associated to hydrothermal vents. Okay? And 60 years later, the field of prebiotic chemistry is a bit more complicated. Okay? But, okay, we have lots of reactions leading to amino acids, to nucleobases, a lot of different nucleobases can be originated. An interesting question is how life took only 5 nucleobases. It's an open issue. Sugar chemistry, the combination of these to form nucleotides and also lipid chemistry. All these together, working together, can account for the proto-metabolism, proto-cell, and proto-genome, the 3 main components of life as I was showing you before. Okay? So, an open issue is the composition of the primordial soup, because every scientist has its own recipe for the primordial soup. You can put the ingredients you want in the primordial soup, okay? In particular, you can think about a very diluted soup, water, some oil, a limited number of components, or you can promote a more compact, rich soup, perhaps with mineral, with surfaces, with a lot of things inside, perhaps more tasty, okay? But the advances of prebiotic chemistry has shown that apart from the reactions that can be produced in bulk solution, the surfaces are very good catalyzers for the formation of molecules. So it's easier to think about primordial chemistry on surfaces that instead on bulk solution. So let's move to a primordial pizza. Yeah? For my Italian friends. And the point is that the best minerals that catalyze reactions are phyllosilicates composed of different layers. So let's move to a primordial lasagna. Okay, I'm very sorry because it's almost lunchtime and these pictures are a bit tempting, but okay, stay here please. So the prebiotic conditions can be as different as you want, okay? And in particular, you can think about a prebiotic soup formed by earth-based compounds or by the delivery of compounds from meteorites or cometary nuclei, as we were listening this morning, okay? Or the combination of both, which is more reasonable. But the point is that, as Jerry Joyce wrote, this dream for a prebiotic chemist, I mean, we can make a high number of compounds using prebiotic conditions, using conditions that could be operating on early Earth, becomes a biologist's nightmare. Because how all these possible monomers were selected to produce the biopolymers? How? When? So, if we want to go from the biomonomers, okay, to Luca and the modern, let's call it modern cellular life, we have a huge gap here, where DNA, RNA, proteins, membranes should have appeared and interact. So we are the major transitions in the origin of life. So we start in physical chemical conditions, okay, early Earth, and we have to go to Luca, and then the diversification of life, okay. In this path, fossils don't help, because fossils only account for cells. We don't have fossils of molecules, and this is the time of molecules, okay? So molecules should have self-organized, should have be able to make a genome and to allow replication, and to form a membrane and to allow a metabolism associated to the transfer of components through this membrane. For that we have, again, these bottom-up and top-down approaches, okay? And there are lots of controversies, of course. This is why this field is so exciting. The first controversy is genetic first or metabolism first scenarios. Okay, genetic first accounts for the origin of coded information and allows you to make schemes towards the genetic self-replicative polymers. And the RNA world is a good example of this scenario. We'll talk about this scenario in the next slides. On the contrary, metabolism first give more importance to the bioenergetic mechanisms that were also needed for the origin of life. Recently, unfortunately, systems approaches have been developed that combine proto-metabolism and genetics and allow the reproduction of the whole system, which is at the end the key for life. You don't have to replicate only the genes, you don't have to make a metabolism, but you have to reproduce the system that contains genome and metabolism. Another open issue is heterotrophic or autotrophic earliest metabolism. If life had an heterotrophic origin, as Oparin said, for instance, the primordial soup should provide organic molecules and energy. Okay? This is the warm little pond of Charles Darwin. So, prebiotic chemistry should be complex, but a simple metabolism could be produced from that. The chemoautotrophic origin says that from CO and CO2 we can go to cells using geological energy source. In this case, okay, the prebiotic scenario is more simple, but it needs a complex metabolism. And the third controversy, I have chosen, there are a lot of them, but these are, in my opinion, the main controversies. The first compartments were cell-like, protocells, or the first compartments were surfaces. Okay, membrane-based compartments allow bioenergetics and the reproduction of the whole system easier, but mineral surfaces are required for chemoautotrophic metabolisms, okay, associated to these hydrothermal vents. These are open controversies, okay? And let's move now to one of the best examples that have been developed during the last 40 years. Originally within this genetic first scenario, but now broadening a lot, which is the RNA world. Okay? First, I would like to show you again the structure of DNA, the double helix, made of deoxyribonucleotides. We like very complex names in biology, deoxyribonucleotides. And the nucleobases can be of these three kinds, C, G, A, and T. In turn, RNA has single strain structure. This is the most stable form for RNA, single strand, not double strand like DNA. The monomers are ribonucleotides because this is ribose, not deoxyribose. It has this OH group at the 2' carbon of the ribose, okay? And the four nucleobases are C, G, A, like in DNA, but uracil instead of thymine, okay? Good, so this is a nucleic acid, a different nucleic acid. In all cases, complementary nucleobases interact through hydrogen bonding, okay? So in this case, although you see this single strand, you can imagine that complementary nucleobases are interacting. When they are available, they interact among them, okay? So in RNA, you can have a sequence. We use the sequence only referring to the nucleobases, so A, G, G, A, G, U, whatever. It falls into the so-called secondary structure by complementary base pairing among nucleobases. And it falls again because, for instance, this part, if these three nucleobases are complementary to this one, they can interact. And they form long-range RNA-RNA interactions. And at the end, they go to tertiary structures. So more similar to proteins in that sense. And what's the interesting point here? Is that RNA, the molecules of RNA combine a sequence so they can take genetic information, like as we will see in RNA virus in viroids, but they have a structure that can perform biochemical functions. So they can work like DNA and they can work like proteins. That's why the RNA is so important. Okay? That's why the RNA world is a prominent model in the origin of life research. It's also interesting in the field of RNA to have in mind that different sequences can fold into the same secondary structures. Because, for instance, to form this simple stem loop structure, we call this a stem loop, different nucleobases can be pairing here. So you have different possible sequences that fold there. Here, this is an example made in the computer in my group, okay? So different sequences fold into this, exactly this one hammerhead. This is called a hammerhead by evident reasons, hammerhead structure, okay? And this is important because this genotype to phenotype degeneration is the technical word we use to describe this difference between the sequence space and the structure space confers robustness and adaptability to RNA. It can allow the presence of different mutations in the sequence because at the end the secondary structure will be the same. So the biochemical function will be the same. So this can be very interesting in the origin of life. By the way, native structure of RNA can be only computed. No, we can see it in the lab. This is one of my papers. And we are using for different kinds of molecules, and in this case this is the same molecule at different magnesium concentration. This is an RNA molecule, okay, 3 nucleotides long. So perhaps it's the first time you see an RNA in your life. Okay, so this is RNA. This is my friends. Okay. If you increase the magnesium concentration, the structure folds by complementary hydrogen bonding. And it folds and folds and folds into a very compact structure. Okay? So RNA structure can be visualized and you can play with RNA structures. And you can imagine how these structures can behave in a prebiotic world. In particular, in this case, this is the best conformation. This is AFM, atomic force microscopy technique, and this is large fields. This is a zoom to a molecule, and this is the 3D representation of this molecule. In this particular case, these are the best magnesium concentration to promote the functional structure of the molecule. Okay, so we can play with this. And this is the flow of genetic information. This is another aspect in favor of RNA. I will show you some of them. As I was telling you before, DNA, okay, replication of RNA, DNA, transcription to RNA and translation to proteins. This is performed by ribosomes. In some viruses, retrotranscription from RNA to DNA, for instance, retroviruses, for instance, HIV virus, and replications of RNA. So RNA plays a central role in the flow of genetic information. RNA also plays a central role in translation, I mean from RNA to proteins in the ribosome. This is a ribosome, this is the crystal structure of a ribosome, and you see here the large subunit of the ribosome. All you see, all these ribbons in white are RNA, okay, folded, okay, very complex structure because it's a quite long molecule, 3,000 nucleotides long, and this is the small subunit. They interact, and here messenger RNA comes, and here you see tRNAs, and here amino acid that is forming the growing chain of a protein. So this is the translation at atomic resolution. And it's interesting that in this part where it's called peptidyl transferase, so when one amino acid joins other amino acid, only RNA is present. It's very interesting. Again, RNA is also important in the transcriptional and translational regulation. This is a quite complex issue, but I don't want to show you so many details, but only to show you this idea. RNA is very important in the regulation of the flow of genetic information, okay? Both in bacteria, eukarya, and eukaryotes. RNA can be also a genetic molecule, as I told you. A number of viruses, and here you have this famous Baltimore classification of viruses. All these viruses have RNA as genetic material, okay? These are DNA viruses, and this is part of the huge amount of different families of viruses that have been discovered in the last years. In fact, if we focused on the main families that infect vertebrates, only vertebrates, okay? These families, Adenoviridae, Herpes viridae, blah, 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 they have double-stranded DNA as genetic material. These families has single-stranded DNA, which is something peculiar in nature, as genetic material. And this is even more interesting. Adenoviridae family that contains, for instance, hepatitis B, okay, has a genome formed by double-strand and single-strand DNA. It's quite complex. But there are more families of viruses with RNA genome, double-stranded RNA genome, and single-stranded RNA genome of different polarities. This is not important now. And here I show you 3 examples of RNA viruses. Okay, so RNA is the genetic material now, in some cases, in some entities. Let's call them replicating entities to avoid this controversy if they are living beings or not. You have Ebola virus, hepatitis C, in this case, and influenza virus. Okay? Yeah, that is common cold. And common cold is also produced by an RNA virus. So all of us are infected, not because of you, but all of us are infected by a huge amount of viruses, and thanks to these viruses we are alive, by the way.
So there are also astroviruses.
Astroviruses, yeah, this is, yeah. It's this family is quite astrobiological, and so perhaps if there is viruses on Mars, this is the first family represented there. So lots of viruses have RNA as genetic material nowadays. And what about the replication of viruses? This is also a complete issue. I just mentioned it in this slide. When more cases of viruses, okay, thanks, because they appeared on the right moment. The viruses you have in your bodies, when they are RNA viruses, every single particle has a different genome, okay? because they mutate at a high mutation rate. They accumulate and they can tolerate a high number of mutations. This is called quasi-species. For instance, your common cold, you are probably in your body, I'm sorry, but you are probably 10 to the 12 different viruses belonging to that species. Different lines. These symbols here represent mutations, okay? And when a viral population replicates, it accommodates more mutations and forms this structure called quasi-species. And this is stable in nature. This is how RNA viruses work. So this is very important because they give viruses a great adaptive potential as cellular parasites. If you are infected by HIV and your doctor recommends you a combination of drugs, the virus will evolve quickly to be resistant to that drugs. So this is a very clever population structure for being an RNA virus. And populations of this kind, I'm not telling that the viruses predated cellular life, but populations of this kind could have been relevant for the origin of life. If we had heterogeneous populations of RNA evolving, that could be very good for the adaptation to the changing environments.
Sorry, why error prone? It means making errors?
Making more errors than DNA. Okay? When your DNA, Homo sapiens DNA replicates, the number of errors accumulated during the replication round is 10 to the minus 9. So one misintroduced nucleotide per 10 to the 9. Okay? In RNA virus, one misintroduced nucleotide per 10 to the 4. So it's a huge mutation rate. That's why viruses evolve so fast. That's why every year we have to get a vaccine against influenza, and that's why people on treatment of HIV, for instance, have to change the drug combinations every 3-4 months. So it's important about the RNA-based system. And about viroids. I told viroids. What are viroids? Viroids are a very nice plant pathogens. The infectious entity here is only an RNA molecule, so no capsid. Okay, so simpler than viruses, of course. This is a naked, meaning without proteins structure, circular, short, from 250 to 400 nucleotides long, and it falls into a compact secondary structure. You can see here one of these viroids that forms a rod-like shape. Okay. This is the representation of the secondary structure, and this is how we see it in our lab, okay? Viruses don't have protein coding sequences. They don't code for proteins. So they use all the proteins that their hosts can provide, okay? But the RNA of a viroid is a template for replication, so they allow heredity, okay? There are two families of viruses up to now, POSP and APSUN. And an example of virus, all of them have very nice names, the potato spindle tuber viroid. Okay? This is the PSTV viroids. So, in this case, it's a replicative entity, of course. It works infecting plants for the time being. No viruses have been discovered infecting animals, for instance. And the genetic material is RNA. So the RNA is working as genetic material in the current biosphere. And there is an inverse proportionality between the genome size of replicative entities, higher eukaryotes like us, lower, I don't like this higher and lower, but okay, eukaryotes, bacteria, double-stranded DNA viruses, single-stranded DNA viruses, RNA viruses, and viroids. So the shorter you have the genome, the more error-prone can be your replication. Okay?
What are the units?
The units are the mutation rate, so the number of misintroduced nucleotides. One every 200 of the nucleotides is not correctly introduced in viruses, and here 10 to the minus 10, so one every 10 to the 10 nucleotides.
And the size?
And the size, the genome size in number of nucleotides of the genome. So our genome is 3 billion nucleotides, one of these. It's not the longest, by the way, but okay, one of these. So we are somewhere here, okay? RNA viruses have typical genomes of 10 to the 4 nucleotides, okay? So the shorter you are, the higher your mutation rate can be.
So what is the difference between lower and higher eukaryotes?
They call lower eukaryotes, for instance, to yeast. But this is not so evolutionary based, that they are lower and we multicellular are higher.
Okay.
Yeah, it's mainly multicellularity, but for me it's not so accurate.
Yesterday when we looked at exoplanets in terms of mass and period, the reasons that there were empty spots in the plot was due to bias, a selection effect. And I'm wondering if there are selection effects here. For example, you have viruses, but the smallest ones are what the mutation rate is 10 minus 2.
Yeah.
Could there be 10 minus 1?
Yeah.
10 to 0?
You are right. Perhaps if
What are the selection effects?
I mean, they are experimental limitations. Okay, and for the time being, the longest viruses that have been analyzed are 400 nucleotides long. So here, perhaps if one day a viroid shows up with 2,000 nucleotides, they would be somewhere here. Perhaps their mutation rate will be lower. So perhaps at the end this is a continuous, but we have some points. And in this representation, they have selected the authors. This is Ricardo Flores' group, a good friend of mine. The authors have chosen a number of dots, not to fool the image, okay? Okay, another proof in favor of RNA, and this is a key question. RNA catalyzes biochemical processes. Okay, let's go back to the flow of genetic information, okay? In this case, this is an eukaryote, and you have the DNA. This is the RNA polymerase that transcribes, and here you have RNA, and this is a ribosome that translates. At the end of the talk, you will be familiar with all this biological stuff. Yeah. And here you have an amino acid, another. So this will be a protein that is finally folding and performing a biological activity. Okay, so this is the flow of genetic information. The interesting discovery in the decade of the 70s was that some genes have parts that are finally represented in the protein, and they are called exons, and parts that are not represented in the protein. So intervening sequences, and they are called introns, okay? And there are a number of reactions called splicing that exist, the intron and ligate the exons, okay? Good. You have, for instance, this scheme here. You have a putative gene with 1, 2, 3, 4 exons and 3 introns, and splicing would take these introns out and join the exons, okay? This is very useful in biology because it allows the differential expression of protein domains, and for instance in our immunoglobulins it is working very actively, okay? In the 70s they thought that all the splicing processes were catalyzed by ribonucleoproteins, so complexes of RNA and proteins, and they're more or less complex. The proteins played the biochemical role, so the proteins of these complexes were the molecules that cut the end of this exon and ligate in this way. This is a topological issue, don't worry about that, and cut this and finally join these two exons, okay? So proteins, ribonucleoproteins, and so on. But a very nice discovery was done in Tom Check's lab in 1980. They were working with this eukaryote, Tetrahymena thermophila, that they knew it had a gene formed by exon 1, exon 2, and 1 intervening sequence, intron. Okay, good. They extracted the nucleus, and from that nucleus discarded the DNA and RNA present in the nucleus and took only the ribonucleoproteins, so the spliceo proteins, okay? And they joined the intron-exon construction and the protein struct of the nucleus in a test tube that contained magnesium, because they knew it was important for the reaction, and GTP is an energy currency that these molecules need. Okay, some molecular biology. There are three possibilities. Okay, you can put the exon, intron, exon construction, okay, in all the experiments, the nuclear extract in 2 of them, and the, let's say, chemical conditions in 2 of them. Here, in the first experiment, with everything, you see the unspliced form of the gene, okay. This is electrophoresis, this is migration proportional to the size of the molecule, okay. So the longer molecule is an unsized form, and here the processed molecule, exon, exon. Okay, perfect. Splicing was working properly. If you don't add magnesium or GTP, reaction doesn't work. So these are required for the reaction. But if you don't add the nuclear extract, the reaction is performed. This is very surprising. What the hell, this band appeared here? You can imagine that they repeated the experiments a number of times. But in the absence of proteins, the intron was able to cut itself and to bind the exons. This is wonderful. This was a discovery telling that in some cases RNA has the information required to perform a biochemical activity in the absence of proteins. Okay? This paper was published in Cell because Nature and Science didn't want to publish it because it was very risky, because it was the first time that the dogma, the biochemical function, the enzymatic function is performed by proteins, was broken. Okay? But Tomczyk was right, and at the end he received the Nobel Prize together with others in few years, by the way. And they called the name ribozyme to refer to this kind of RNAs that perform enzymatic activities. So these are ribozymes. This is the process of the intron. This is the secondary structure of the intron, okay, RNA secondary structure, you are familiar with this. This is the tertiary structure, and this is a nice crystal structure of the intron. So this is the first ribozyme that was discovered. So you have here in front of you a catalytic RNA. Okay? There are 2 types. I will not go into details. Type 2 self-splicing introns is longer and more complex and so on. And almost simultaneously a different ribozyme was discovered. The group of Sidney Almond was working with the precursor of tRNA, transfer RNAs, that will accommodate the amino acid here and interact with the ribosome to perform the translation. They need to be cut at this site, at this end of the molecule, and the molecule that cuts is in fact a ribonucleoprotein composed of an RNA motif and a small protein. Okay, Almond discovered that the catalytic function inside this ribonucleoprotein complex was performed by RNA. So RNase P was the second ribozyme discovered. It's interesting that we know some ribozymes that are operating inside our bodies, so we are alive thanks to the ribozymes, among other aspects, of course. And here you have the one of the 2 types of self-splicing introns. These are 3 examples of short ribozymes that are operating in nature. And as I told you before, the peptidyl transferase domain of the ribosome is also a ribozyme, because this part of the ribosome, we were talking about that yesterday, is entirely formed by RNA. And other RNA enzymes, so ribozymes, have been produced by in vitro evolution. I will tell you something about in vitro evolution. So RNA can have a number of catalytic activities. So Tomczyk, once the ribozymes were accepted by the community, and he and Alma received the Nobel Prize, of course, wrote hundreds of papers, as you can imagine, and one of them shows this very interesting cartoon about the possible evolution of the catalytic function. So perhaps at the beginning there were ribozymes, or ribozymes interacting with peptides, why not? These peptides being formed abiotically in the absence of ribosomes for the time being. This would have produced ribonucleoprotein enzymes that could have evolved to new functions performed by these ribonucleoprotein aggregates or to protein enzymes. And in our current biosphere, almost all the catalytic activities are performed by protein enzymes. Okay? Some words about in vitro evolution of RNA. Going back to 1967, this very elegant scientist, as you see, Sol Spiegelman, asked himself if evolution could proceed in the absence of cells, what is a quite relevant question, and even in the absence of viruses. So, can molecules evolve inside the test tube? It's interesting. What he did, he took one virus that infects bacteria. There are lots of viruses that infect bacteria, as you can imagine. He took its genome. It's a 4,000 nucleotides genome. Here you see a picture of it. And okay, he put inside a test tube the genome, the protein coded by the viruses that replicated this genome, and some salts and the ribonucleotides needed, and so on. The genome replicated, okay, more copies of the genome. When these ribonucleotides and salts were exhausted, it was transferred to a new test tube, and again and again and again and again. Okay, a nice experiment, and these were the results. As expected, an RNA of the length of the complete genome was accumulating during the process. Perfect. But another molecule replicated faster and faster and faster, and they analyzed it, and they found that it was shorter and shorter and shorter. So this progressively shorter RNA molecule derived from the viral genome, so a part of this was produced because the evolution inside the test tube. So molecules were evolving inside the test tube. If you allow RNA molecules to evolve, they can evolve, and this is quite interesting. Here, what was the selective pressure? Because you need selective pressure to evolve. You should replicate fast. This is the selective pressure. So the faster you replicate, the higher amount of concentration you will have.
To be sure, duplicating is the same thing as replicating, or is it different? Duplicating, because it's
replicating, isn't it? Yeah. Duplicating, because one molecule gives 2, 4.
But the same molecules, they're exactly the same.
In this case, yes. The protein was bound to the molecule and copied the whole molecule. Okay. But the interesting point was that even shorter versions accumulated. Okay, this is a very interesting discovery, but the technology was not available. I mean, these are the initial era of molecular biology to go in deep in this kind of experiments until 1990. Okay, in 1990, two groups, Larry Gold and Jack Szostak, simultaneously published a couple of very interesting papers. In the first one, Twerk and Gold coined presented this experimental setup I will tell you about, called systematic evolution of ligands by exponential enrichment, or SELEX. And Jack Szostak coined the term aptamers, okay? From the Latin aptus, so fitted, and the Greek meros, unit. What is this? This is a modification of what we did. You can start with an initial population of RNA molecules, very large, let's say 10 to the 15 different molecules, okay? You put it in contact with a ligand, okay? This ligand, and all the molecules can adopt different tertiary structures, and in some cases the structure could be compatible with the binding to this ligand, okay? If you are bound to the ligand, you remain. If not, you are discarded, okay? Once you have been selected, you are amplified. In this case, you need a reaction called reverse transcription that gives cDNA, don't go into details, then PCR amplification, and then transcription, and then again you have a new starting population that is submitted to this selection step, and again and again and again a number of rounds until all the population is able to bind the ligand. And this works perfectly. Okay? So these Selex-based experiments are giving rise to dozens and hundreds of aptamers. We are working in my lab with different aptamers, and this is fascinating. In 1 month you have an RNA molecule able to bind to whatever you want, from small molecules to cells, for instance. And okay, you have here some examples. In these 3 panels, the aptamer is the large part of the drawing. This is the ligand, adenosine in this case, and this is the aptamer that binds the ligand. This is the aptamer that binds adenosine monophosphate, and this is the ligand, the aptamer that binds the red part, which is ATP. In other cases, the ligand itself is larger than the aptamer. For instance, this is a protein, and this is the aptamer that binds here. That binds because it has been selected in vitro.
Aptamers are RNA?
They can be RNA and also single-stranded DNA. Okay? But these examples are RNA. Are aptamers in nature? No. Aptamers are the produce of the Selex experiments, so in vitro evolution of nucleic acids. But in nature there are some genes that contain at one of the sites an RNA structure that is able to bind to ligands. Okay? They are called riboswitches. And riboswitches are considered the natural form of aptamers. Okay? I will not go into details, but I think this is important. So, a number of different evidences from different fields. And at the end, the idea is, if you look at the flow of genetic information and you ask yourself, what came first in the origin of life? DNA or proteins? You have a very nice paradox, because DNA is required to make proteins, but all these reactions are performed by proteins. Okay, in this case the ribosome is a combination of proteins and RNA. So if you see this one, you have a topological problem. Let's say, what came first, DNA or proteins? Molecular biologists, we are not very clever, so instead of trying to solve this puzzle, we accumulated proofs coming from different fields. And okay, as I showed you, the information, okay, in all cells is stored in DNA, but in viruses and viruses in RNA. Okay, some viruses and all viruses and viroids. Proteins don't take information, don't harbor information, are the product of information. What about the structure? The structure in all the machinery of life is done by proteins, let's say, and in some cases by RNA. For instance, a large part of the ribosome structure is formed by RNA. And what about catalysis? In most of the cases, the catalytic molecules are proteins, but ribosomes are current examples of catalytic RNA. So perhaps the answer to this conundrum is RNA. Perhaps RNA came first. And this is the basis of RNA world model. Okay? So, RNA can perform a lot of activities. There are extra proofs about the antiquity of RNA when you compare it with the DNA. For instance, the deoxyribonucleotides that form the DNA are formed in the metabolism after you have a ribonucleotide. So the metabolism has a key to the antiquity of RNA. Okay? Also, the main energy currencies, molecules such as ATP or GTP, are derived from ribonucleotides. And these are used by all cells in the planet. And several coenzymes like these ones are also derived from ribonucleotides. And if you have in mind the flow of genetic information, RNA must be also prior to proteins, because protein biosynthesis is dependent, fully dependent on RNA. So these are a number of proofs in favor of the antiquity of RNA. So we have all the ingredients for the RNA world model that says that RNA is the only known macromolecule that can function as genotype and phenotype. Sequence of the molecule and structure and function of the molecule. thus allowing evolutionary processes at the molecular level. So it can evolve because the same molecule has genotype where mutations can accumulate and phenotype that can be selected by evolution. Okay? So an RNA wall could have existed as an intermediate step between prebiochemistry and cellular life. This model was published by Walter Gilbert in a short article in Nature in 1986, and here it says, one can contemplate an RNA world containing only RNA molecules that serve to catalyze the synthesis of themselves. Okay, current versions of the RNA world don't say that only RNA molecules. Okay, RNA molecules plus perhaps peptides, plus low molecular weight compounds, plus, but without proteins and without DNA. So RNA plus a number of small molecular weight molecules can serve to catalyze the synthesis of themselves. Indeed, there are very good groups working in the integration of the so-called ribosite. This is a model of how a cell could have existed with only RNA working as the genome and as small parts of the RNA performing biochemical functions, ribozymes. So Jack Szostak's lab is a pioneer in this kind of work, together with Pierluigi Luisi in Italy, and David Wimmer in the States also. And they have this kind of interesting models of the replication of the whole system. So they are working towards the experimental design of a possible ribosite that could resemble this pre-cellular world. So, okay, we are able to fill a little bit this gap. Okay, we had this gap, and 40 years of research in molecular biology tells us that, okay, at the beginning perhaps RNA replicated itself and performed biochemical functions. The proteins were synthesized via peptide transferase activity, and proteins were much better performing biochemical functions, so the catalytic activities were moved towards proteins, and then some proteins were able to retrotranscribe RNA to DNA, and DNA showed that it was a molecule better than RNA for storing genetic information. So at the end, in our cells, the genetic information is stored in the form of DNA. Most of the biochemical functions are performed by proteins, and RNA is an intermediate protein, but RNA was probably the first one. So this is the basis of the model. And okay, is it possible to synthesize the RNA? So okay, we say that RNA is very important for the origin of life, but could it be synthesized in primordial conditions? I will not go into details, but it's interesting that in the last 30 years, lots of researchers have been trying to, in the field of prebiotic chemistry, to combine ribonucleotides joining ribose and nucleobases, okay? But the point is that this traditional way, in blue, is not effective. So you cannot make a ribonucleotide as expected, because even if you see a molecule now in nature and you say, okay, a ribose, a nucleobase, and here the phosphate, finally, okay, let's make it by forming the nucleobase, then the ribose, then the phosphate, and combine it as a puzzle. Okay, perhaps nature didn't perform the synthetic process in that way. And the group of John Sutherland was able to show in 2009 that using these 4 small molecules, and here you have the whole pathway, you can go to nucleotides very efficiently. So this is a prebiotic model for the synthesis of ribonucleotides. So, okay, ribonucleotides could have been synthesized in the primordial world. Could they polymerize? Okay, because if you have a ribonucleotide, you need to polymerize to form RNA molecules. The polymerization of ribonucleotides always requires an activation of the nucleotides. It's a chemical issue. In nature, ribonucleotides are activated by three groups of phosphate. Two of them are stripped away when the ribonucleotides interact. And in chemistry, we use this imidazole group to activate the nucleotides. Okay. Does this kind of monomer interact with others once it is activated? In bulk aqueous solution, this process is not very efficient because the reaction is shifted to this to the decomposition instead of the polymerization. Okay. So only the dimers or tetramers of RNA are formed. Okay, experimentally. To reverse the direction, you can use thermodynamics, so use water-poor media because the condensation reactions require water removal, and you can use environments that promote nucleotide stacking. Okay, these are technical issues. And to improve the rates, so kinetics, you can use catalysts, mineral surfaces, metal ions, heat, etc., etc. Among mineral surfaces, this is the seminal work of Jim Ferris and others. They use suspensors of minerals in water or salt precipitates. Some of them have these surfaces and interlayers, like the primordial pizza and lasagna, as I told you, that are able to concentrate, order, catalyze the formation of RNA chains and also to protect the chains from hydrolysis. The best up to now is monomerilionite clay. Okay. So this clay is able to form oligoribonucleotides, so short chains of RNA. Other scientists have used lipid bilayer matrices because dried films are promoted at the interaction between water and the lipid phase, and they are able, I mean, with this system, you can obtain 50 mers with this system up to 30 mers. And a new option is to use the eutectic phase in waterized medium. That's why I asked Daniele the possibility of having ice in primitive Earth, on primitive Earth, because these microenvironments concentrate the substances at the surface and the interlayer between ice and water. And okay, in this system they are able to get to up to 22 mers of RNA. So RNA can be polymerized. If you polymerize RNA, for instance, this is a clay monomerilionite, okay, so activated nucleotides, and okay, again, electrophoresis. This is a 10 mer, 11 mer, 12 mer, up to 40 mer of RNA. Could this process lead to the appearance of the first RNA able to copy itself? There are some groups, and this comes from Holliger's group. They are trying to evolve in vitro an RNA molecule able to catalyze its own replication. This doesn't exist in nature, but if one RNA would be able to replicate itself, that could be perhaps close to the origin of life. Can we go from this random accumulation of nucleotides up to 40 more or less to a complex molecule like this? I will not go into details, but we have been working in models that can allow this, because when you fold the RNA oligomers, some of them can adopt a hairpin-like structure, and hairpin is one of the kinds of ribozymes in nature, and hairpins are able to join different RNAs. So you produce short oligomers, you combine them, and you make the puzzle that can allow you go to the final polymerase. Okay? By the way, when we are thinking about ribosome able to catalyze its own replication, we have to compare it with the situation in which a protein catalyzes RNA replication. For instance, during the infection of a cell by a virus. Here you have an RNA template. You use monomers. You can think about the Schultz-Spiegelman experiment I showed you before. Okay, the template, the genome, the monomers, and there is a protein that performs a catalytic activity. It is not affected after the process, but at the end, using these monomers, it performs a template-dependent replication, and 2 molecules are originated, okay, with some mutations. Okay, look at the image, and now let's move to ribozymes. Exactly the same, but a ribozyme with the same sequence than the template would produce 2 copies of the template, and the catalyst will be unaffected. So there are groups trying to get this molecule that can act simultaneously as catalyst and template. This is a key issue. As our friend Antonio Lazcano says, he cannot be here, but I would like to remind him, the catalytic, regulatory and structural properties of RNA combined with their ubiquity in cellular processes are consistent with the proposal that they play a key role in the early evolution and perhaps even in the origin of life. But even if this possibility is not accepted, because there are some people, as you can imagine, that don't accept the RNA world model, the origin and manifold roles of RNA in extant biology need to be addressed. So it's a very nice sentence. By the way, was the RNA the first genetic polymer? I am about to finish, don't worry. Muriel told me from 2 hours to 2.5 because it's the last one. So, but I will try to finish in 10 minutes, the most. So RNA was probably prior to DNA, but was RNA the first genetic material? Probably not. Different genetic, let's call artificial genetic molecules, so polymers with different backbones from which nucleobases protrude, okay, in all cases. B is here nucleobase, and they are called GNA, pRNA, TNA, this is treose nucleic acid, locked nucleic acid, peptide nucleic acid, etc., etc. They have the advantage of being more stable than RNA, so perhaps they were good candidates to have preceded RNA, and they are compatible with RNA in the sense that these structures have been designed to be at the right position and distances, the nucleobases that allow interacting with RNA or DNA. Okay? Are they prebiotically plausible? This is an open issue. So, is it possible to have monomers of DNA? Who knows? Okay? They are part of the groups working in this field, asking themselves if they are prebiotically plausible. Here, only one example. This is RNA and this is PNA, peptide nucleic acid. One of the possible, let's say, precursors of RNA. If you see, this chain is more similar to a protein chain than to a nucleic acid backbone. No ribose, no phosphate. Okay? But it has nucleobases at the right position that allow them to interact with RNA and even to form the helices. Okay? So, could some of these polymers be present in early life, early Earth? We don't know. And after all this evolution of the RNA world model, we are now facing an epoch in which we like to integrate RNA world with metabolism, with compartment in a systems framework. This is what we are going to talk in the conference in 3 or 4 days. Because we don't have to forget that the life, as I told you at the beginning of my talk, is the combination of genome, metabolism, and compartment. And only the combination of these 3 elements gives rise to a ternary system capable of undergoing Darwinian evolution. So, if RNA world is only concentrated in the origin of the genome, okay, it doesn't account for the complexity of life. This is one model we have. I will tell you about it in a couple of days. So, at the end, if everything, as Darwin, okay? I think that if everything we have been talking about in this presentation is true, perhaps after the prebiotic chemistry and before the cells, the gap we had can be filled by different steps. I will not go into details of this scheme, but probably RNA analogs came first, then RNA, the first ribocytes, so cells containing RNA, genetic code, ribosomes allowed the advent of proteins, so ribocytes with RNA plus proteins, compartment, metabolism, replication, integration, which is essential for life, as I told you. Perhaps this is the origin of life. Perhaps in these systems, able to replicate themselves to reproduce themselves are the basis of life. The development of a complex metabolism, perhaps the appearance of a DNA molecule that, as I told you, showed a more active role in keeping biological information, genetic information, and then modern cells. So perhaps this is how it worked. This could be a timeline, okay? So this is the first picture I showed you, and this is the time where fossils of bacteria, wherever they are present. So these 350 million years could have accommodated this process, or perhaps it was shorter, we don't know. Perhaps RNA viruses were originated once the first ribocytes were present, and DNA viruses, the first of them, were present once DNA-based systems were present. A final question, almost final. More than one origin? Why not? If we have here the physico-chemical conditions on early Earth, we know that we have been talking about this trunk of the tree of life. We are right to Luca and bacteria, eukarya, archaea. Okay, but could this have happened? Why not? Why not? The Earth, remember, was a huge laboratory with different environments, with different physico-chemical restrictions. Perhaps some experiments towards life were initiated, but sometime later they disappeared because they didn't get the right nutrients, or because a meteorite finished the life in this warm little pond, or whatever. So perhaps, as this is part of a chapter coordinated by my friend Puri, where I also contributed, in Origine et Histoire de la Vie. Perhaps there are some dead ends in evolution from the chimie prebiotic, in this case, that at the end went to a cell or group of cells that would be Luca, that were strong enough to initiate a Darwinian evolution that has continued up to now. Okay, but these dead ends could have existed in nature. Okay. By the way, what do we know about Luca? Okay, Luca, only a couple of slides about it. To go to Luca, we have to use the top-down approach. So what you do is you get the genetic information present in all archaea, bacteria, and eukarya, compare it, and say, okay, the shared genes could have been present in the ancestor. Okay, if you go to the shared genes among archaea, bacteria, and eukarya, you see that there is a core group of about 50 to 70 genes that perhaps they were key for the earlier life, and almost universal, there are about 600 genes. Okay, so coming back to this scheme, Luca could be something like this. We don't know, but perhaps it had a genome here in this picture. It says DNA or RNA. Okay, most scientists would say that DNA, genome, transcription machinery, translation machinery, the basic central metabolism which is maintained and is common to all living beings, something, a TPase machinery that allowed the bioenergetics which is required, and some transporters and proteins associated to proteins. Perhaps with this gene set you have everything you need to start Darwinian evolution, because at the end it was evolution. Okay, perhaps the most fascinating process in the history of the universe. I like very much this, I am about to end, and this definition of Lecointre and Goujon in our Muriel, this is especially for you, in our Encyclopedia for Astrobiology. That is a must in the field. It's something all of us should read every year, let's say, because it's quite... they have a very nice entry about evolution in this encyclopedia. And they say, in biology, evolution is the change of organisms through their variation, then transmission of this variation to their progeny, and eventually selection of some phenotypic variant or trait in the long run as a result of environmental conditions. So, mutations in the genome, selection of the phenotype. This is the basis of evolution, and this has been working at least since Luca appeared on Earth. But usually, evolution is not directly accessible to our senses, since large animals have long generation times and require very long periods to exhibit spectacular changes or speciation events. That's why if you want to play with evolution, if you want to see evolution in front of your eyes, you have to use bacteria or viruses or molecules. Okay? But remember, this sentence is not clear if it was said by Darwin or not, but it's very nice. It is not the strongest of the species that survives, nor the most intelligent, but the most responsive to change. That's a great sentence. Perhaps it was Charlie who coined it, but if not, it's also very interesting. So evolution is working, okay? And as Theodosius Trzaskey wrote, nothing in biology makes sense except in the light of evolution. Two final examples of evolution. This is related to the previous talk that I introduced after your talk, because I think it's interesting. If you see here a picture of the evolution of our atmosphere, don't trust too much to these shapes and so on, but okay. From this perhaps primordial atmosphere to this one, we can discuss about that. The oxygen photosynthesis performed by cyanobacteria that perhaps appear early, but here was definitely present, produced oxygen. Oxygen was accumulating in the oceans. It formed the binded iron formations because it oxidized all the reduced compounds, and then it started accumulating in the atmosphere. Oxygen is a demon, it's a poisonous gas, because life had evolved since here to here in anaerobic conditions. So organisms had to react to this new selective pressure that has been produced by nature also. So they have two possibilities: to hide in anaerobic microenvironments, for instance in the deep subsurface, or to use this toxic gas. That's why evolution selected a new kind of metabolism called aerobic respiration. Let's use this poisonous gas to accept the electrons at the end of the metabolic pathway. So this is a, I think, very nice example of the co-evolution between life and the planet that harbors life. Okay? Perhaps something like this is happening beyond Earth, we don't know. And it's also interesting to recall only on a slide that once life originated, has been able to adapt to different physical-chemical environments. And you have a good number of examples of extremophilic environments such as thermophiles. This is an hydrothermal thermal vent, and here you have extremophiles surrounding this hot spring. Cryophiles, also called psychrophiles in Antarctica, or acidophiles. This is the Tinto River in Spain. Okay. After evolution, okay, this has been completed. We are the final product, the current biosphere, of at least 3.5 billion years of biological evolution. This is the last universal tree published so far, and all these you have here are bacteria. These are archaea, and these are eukaryotes. Part of the eukaryotes are unicellular also, so almost 85% of life on Earth is unicellular. So this is the planet of microorganisms, okay? And we are here in one of the branches, nothing else. And we are here in the middle of this amazing biodiversity. And this is everything I wanted to tell you today. Thank you very much.